Hello everyone, in this episode we're going to talk about another type of neurological disease, that is a Canavan disease. It is reported that Canavan disease is highly frequent happen in the Jewish population. To be specific, it happened among the Ashkenazi Jewish population. But in Malaysia, we receive no data and there is no case has been reported about this Canavan disease. But anyhow, it is very important to know about the basic knowledge or basic background about the Canavan disease in educational way to educate people around the world about this disease. Okay, now we're going to look more into the Canavan disease and we're going to understand about one thing, that is about the axon and the myelin sheath that covered the axon. Here I have two models of the axon. The first is axon with the healthy skin of myelin sheath, and the second one is the axon with the sick myelin sheath. What happened to this axon with the sick myelin sheath? Of course, it will affect electrical impulses to be sent from the sending neuron to the receiving neuron. So, what is the function of the myelin sheath? The first function of the myelin sheath is to ensure that electrical message that is transferred from the soma to the terminal button is sent in accordingly in its place, so that when it travels along the path from the axon to the terminal button, it's not running away, it's happened in a place. And secondly, the function of myelin sheath is not just to cover the axon, it also helps to speed up the rate of electrical transportation or electrical conduction inside the axon. If the myelin sheath is damaged and happen the malformation of the myelin sheath, it will affect the speed rate of the electrical. It might happen to become like electrical message should be sent very slowly, or maybe it not happen at all. Now we have to understand why myelin sheath become like this. It is because of one of the enzyme inside of our brain. The name is aspartate enzyme that affect another type of acid in the brain that will affect the formation of healthy myelin sheath. With the deficiency of the aspartate enzyme in the brain, it will affect the NAA, that is aspartic acid, and at the same time, it will affect the formation of the myelin sheath. And therefore, the deficiency of this enzyme that cause the deficiency of the quality of the myelin sheath.
Canavan disease is a progressive fatal neurological disorder that begins in infancy. It is caused by a deficiency in the genes encoding a protein called aspartate. Aspartate is needed to break an acyl L-aspartate, or we call as the NAA. NAA is a molecule that is thought to be involved in the removal of water from a certain type of brain cell, as well as in the formation of our myelin lipids. Then, what happens if NAA is not broken down? The excess of level of NAA lead to an increase in the osmotic pressure in certain type of brain cell, and it causes demyelination. Thereby, it preventing the proper transmission of our nerve signal.
Symptoms of Canavan disease varies, but in general, it usually begins in early infancy, commonly by the age of 3 to 5 months old. Infants from Canavan disease suffered from rapid increasing head circumference, lack of head control, and abnormal muscle tone such as stiffness and floppiness. Not to mention, delay in motor skills such as turning over or sitting without support. Yet another symptoms of Canavan disease take place in the form of paralysis, blindness, or hearing loss. Feeding and swallowing for patients with Canavan disease could be challenging at times. Furthermore, seizure and sleep disturbance may take place most of the time. Therefore, we could say that in general, The symptoms for Canavan disease happen in enormous and various ways.
There are many tests that may be performed to diagnose the Canavan disease. For example, now we have CT scan or MRI scan. It is very useful to look for the degeneration of the white matter in our brain. Another test that may be performed to diagnose the Canavan disease is urine and blood fluid test, which is we look for increased N-acetyl L-aspartate or NAA in this fluid. Canavan disease also can be diagnosed by using genetic testing for aspartosylase genes mutation. Since the genetic basis of the disease is known to be a defect in genes encoding or we call as the NAA, then the DNA of the child can be tested to look for mutation in this gene. Yes, parental genetic diagnosis is possible because genetic counseling is recommended for prospective parents who have a family history of Canavan disease. Parents also can be screened to see if they are carrier of the disease. Okay. First, we must understand that Canavan disease is an autosomal recessive condition, which is a patient has 2 copies of infected genes. Then, who is the carrier? And the carrier actually those who has only 1 mutated genes, which means that the genes doesn't give any impact of the disease because another genes is still functioning very well to produce the necessary enzyme.
When it comes to special treatment, the scientists couldn't find any special treatment for the Canavan disease. But anyhow, the treatment is still given in a way of symptomatic and supportive. The medications are given to support the nutrition and hydration so that the seizures can be controlled and avoided.
One of the most devastating news about Canavan disease lies in the fact that the prognosis for Canavan disease is very poor. Death usually occurs before the age of 10, yet some might survive until their teens and 20s.
We know that researchers couldn't find one special treatment for this disease, but we believe with the advanced technologies and facilities that we have today, in the future we could find one significant treatment for this Canavan disease.